Hello dear students, today we will discuss an important topic, chromosomal mutation, as prescribed in the syllabus of Genetics and Plant Breeding for 6th semester BSc. Before going to the chromosomal mutation, first of all we will study what a mutation is. So, a sudden appearance of marked heritable variation in the nature of any organism is called a mutation. According to Darwin, this is a sudden appearance of remarkable heritable variation in the sequence of the genome in any organism. Now, the individual undergoes a changed phenotype. According to Bateson, mutation is discontinuous variation. According to Hugo de Veres, it is a drastic heritable change which cannot be traced. According to Sinnott, it is any heritable change in genotype. These changes are passed further generation to generation and hence these are permanent changes. Some common examples we meet are sudden appearance in the change of flowers of Oenothera limaciana, an ancon sheep with short legs, white-eyed Drosophila melanogaster between the red-eyed Drosophilas, sudden appearance of hemophilia disease in the royal family of Queen Victoria. The process by which mutation appear is called mutagenesis. There are many factors which cause mutagenesis. One is a natural mutagenesis or other is the induced mutation. Natural mutation is also called spontaneous mutation. This appears in an organism without the interference of external environment. Means this type of mutation is actually due to the internal factors of the organism like error in DNA replication or error during crossing over or some convulsions in genes and chromosomes during replications, etc. Whereas, on the other hand, induced or artificial mutations are bring about exogenously through some agents which are called mutagens. The agents which cause mutation are actually called mutagens. Some mutagens are temperature, physical mutagens like alpha, beta, gamma, UV rays, X rays, electromagnetic rays. Some chemical mutagens like mustard gas, urethane, formaldehyde, some DES, EMS, MMS, hydrazine, nitrous oxide, etc. The full form of DES for your convenience is diethyl sulfonate, EMS ethyl methyl sulfonate, MMS methyl methyl sulfonate. Some age factors are also there which cause artificial mutations. The general characters of mutations are generally recessive, means these are due to the deletion of some dominant genes, hence these mutations are recessive, although some dominant mutations are also seen. Secondly, these are harmful to organisms. These are random in appearance, means occur at any time at any part of the cell. And last, mutations are recurrent, means a same mutation can occur again and again. Now, what's the role of mutation? It is an ultimate source of our genetic variation. It leads to evolution, it leads to the formation of alleles of genes and helps in adaptation of organisms with the environment. Now, the types of mutations are number 1, gene mutation, the mutation which appear in genes. Second is chromosomal mutation or the mutation in the structure of chromosome. And third is genomatic mutation, which is also called numerical mutation. It is a change in basic number of chromosome, hence it is also a kind of chromosomal mutation. Now coming to the point of chromosomal mutation, first of all we will describe the types of chromosomal mutations. As we can see, we have divided it into two parts, A and B. The A is The type of chromosomal mutation which is due to the change in structure of the chromosome or the change in architecture of the chromosome. The other is the numerical mutation which is due to the change in number of chromosome in a genome. The structural chromosomal mutation can be divided again into two parts. Number one, change in number of genes and change in gene arrangement. This change in number of genes is of two types, deletion and duplication. The change in arrangement of genes is of two types, inversion and translocation. Here, the numerical change in chromosome is of again two types, aneuploidy and euploidy. So, the mutation which is involving either a whole chromosome or a portion of a chromosome is called a chromosomal mutation or a chromosomal aberration. Now, we will discuss about the first topic, deletion. Now, the first type of chromosomal mutation is called the deletion. As we can observe here, a chromosome having its two arms and has a central centromere. Now, as we all know that genes are arranged in a linear manner on a chromosome. These A, B, C, D up to J, these are the genes which are arranged in a linear manner. The position of each gene is fixed, and when a chromosome breaks into several pieces and unites again or reunites, some part or fragment gets lost. This type of losing a segment or a portion of chromosome is called deletion. This is also called deficiency mutation. This is an irreversible process and cannot be healed back. Deletion is of two types, terminal deletion and intercalary deletion. If a sequence of genes are lost from the terminal ends of the arms, either from this or from this arm, from the terminals, then it will be called a terminal deletion. Example, in this chromosome ABCDEF, the AB part has got lost and only CD and EF are left now. So the terminal part of the arm is being lost, and such a type of deficiency or mutation is called terminal deletion. If any interstitial part of a chromosome, means the genes lying in between the portions of terminals and centromeres are lost, that is called intercalary deletion or interstitial deletion. Here, first of all, a loop is formed. Now this loop is cut by this end and this end. This forms a ring like this. C, D, E are lost in the form of rings. And this B unites with the F. Adjacent, it comes close to the F. And now the portion left is A, B, F instead of A, B, C, D, E, F. Now this is called intercalary deletion or interstitial deletion, as it is a loss of interstitial segment of the chromosome. Now, what are the genetic effects or the cytological effects of deletion? Since deletion leads to the loss of some important genetic informations or the genes, hence it is very hazardous to the organisms. Some unique type of phenotypes are obtained due to deletion. Example, notched wings in Drosophila, Cry-Duchet syndrome in human beings. This is the deletion of a segment from the 5th chromosome of the human being. Turner syndrome in human beings. These are some examples which occurred due to the deletion of some segments from the chromosomes. In lower organisms like Chlamydomonas, yeast, fungi, this deletion is usually lethal, means the individuals die away due to the deletion of the segments of chromosomes. Now the two phenomena that can be seen due to the deletion are recessive lethality and pseudodominance. These are the two consequences of the deletion. Recessive lethality means due to the loss of the genes, the individuals become recessive lethals. And pseudodominance means if it occurs in the chromosomes, if some genes are lost which are dominant, then the recessive genes start acting as a dominant ones and they get expressed in the phenotype of the next generation. Hence, such type of expression is called pseudodominance. The cytological effect of deletion is we can observe under microscope is formation of centric and acentric chromosomes in eukaryotes during mitosis due to deletion of segments of chromosomes. And second phenomena which we observe under microscope is buckling or loop effect. This is found during synapse of heterozygotes and it was first observed by Stanfield 1986. He found that due to the deficiency of some genes during the pairing, one of the chromosome forms a loop since it has B, C, D extra genes which are lost in this A and E, between this A and E. Hence this A which will be paired with this A and E with this E. The rest of the genes or the part of segment or chromosome will form a loop which can be observed during the crossing overs. So this was about the deletion. In the next lecture, we will learn about duplication. Thanks.